Great. So can we start? Let's go ahead.
Of course.
Okay, wonderful. So hi. First of all, thanks a lot for the opportunity and for inviting me to present our results to you. I'm very excited to do so today. So I'm Ofir. I'm a PhD student under the supervision of Professor Ayelet Landau at the Hebrew University of Jerusalem. And I wanted to tell you about the neural signature we found for accumulated evidence underlying temporal decisions. So I'll start with a word about temporal decisions. So often in our daily lives, we have to make some sort of temporal decisions, and typically we can phrase those questions as, has it been long enough since something has happened? So for example, let's say you're cooking popcorn on your stovetop, and at the end of the process, you're beginning to ask yourself when it's time to take the pot off the fire. And this question can also be phrased as, has it been long enough since I heard the last kernel of corn popping that I know that the rate is low enough and I can simply remove the pot from the fire? Now, we are interested in the cognitive processes that underlie such temporal decisions. And today I want to show you a way we found using EEG to study these cognitive processes. And the task we used is the temporal bisection task, which is a very commonly used task in timing and timing perception research. And this task is divided into two parts. The first part is a training part, and then we move on to the main block. So the training part of the bisection task is designed to familiarize the subjects with two durations, a short reference and a long reference. And the procedure goes something like this. So we present the subject with a stimulus, let's say a half-second long stimulus, and we simply ask our subject to respond whether they thought this stimulus was short or long. So let's say it's the first stimulus and they just guessed this was long. Then we give feedback to the subjects, letting them know that they were incorrect, and we move on to the next trial. In the next trial, let's say we present a new stimulus, this time 2.1 seconds long. Now, this time, the subjects correctly identified this one as a long stimulus, and we give them a correct feedback. This goes on for about 10 or 12 tries, after which the subject is familiarized with the short and the long reference durations of the task. And now, after training, we move on to the main block, and the main block is simply showing the subject with stimuli anywhere between the short and the long reference duration, and each time we ask the subject to respond whether they thought the stimulus was more similar to the short reference or to the long reference. Now, at an overview, we can see that the training block then simply contains the short and long reference, and the main block contains both these references as well as a bunch of intervals in between. Now, if we think about a single trial in this task, we can ask ourselves, when do we look for timing processes during this time? So let's say we have a stimulus and a response of the subject. The first process of interest is an ongoing timer, some sort of timer that tells the subject how long it's been since the stimulus started. The second process is some decision process that has to end before the response, but can start before the stimulus is over, during which the subject can decide whether this stimulus is long or short. And we focus at the offset of the stimulus for two reasons. First, this is the first moment at which the subject can know for sure whether the stimulus they were presented has a specific duration. So that's the first moment the subject knows the specific duration that was presented. The second reason is that this is the latest point at which the subject can start making their decisions. So this is like a transitional point in the task, and we will look at the EEG surrounding the stimulus offset. So let's start with the results of the first experiment, which was a visual experiment. We used visual ratings of stimuli, and the intervals we tested in this design were between half a second and 2.1 seconds. So let's start with taking a look at the EEG to the shortest duration in this block, which was a half a second long stimulus. On the x-axis, we have the time from stimulus offset, where 0 marks the offset of the stimulus. On the y-axis, we have the potential in microvolts. And we see a strong positive potential following the offset of the stimulus, for which we also have the topography of potential across the skull, which is centered in the center of the head with a frontal bias. Now let's add another stimulus to the mix, the second shortest stimulus in the block, which was 0.7 seconds long. And we basically see the same ERP pattern, but we have a smaller positive potential. Now let's take a look at another stimulus, the third shortest stimulus in the block, 0.9 seconds. This time we have basically the same response, but an even smaller positive potential to the offset. And this goes on. As we take a look at longer intervals, the positive potential decreases. And if we add all the intervals we used in this block to the plot here, we can see that the potential has a systematic dependence on the duration of the stimulus. At first, it decreases in what seems to be a linear fashion. And then for the longer intervals in the block, it seems to saturate at some potential level. Now, to summarize this response into a single number, we took the average across a window of 300 to 500 milliseconds post-offset. Let's add this to a graph, which is in the center here. So we have the maximal response to the shortest stimulus, a linear decrease, then an elbow and a saturation, such that the responses to the longer stimuli in the block are the smallest. But this is only one measure of the output of subjects in this task. The other measure is, of course, their behavior. So in a bisection task, we typically look at the psychrometric curves. That is, on the x-axis, we have the stimulus duration, and on the y-axis, we have the percent of trials at which subject categorize this specific duration as long. And we get the typical shape. For the shortest durations, subjects always say short. For the longest duration, they almost always say long, and a smooth transition in between. Now, if we take a look at both of these outputs, the behavior on the one hand and EEG on the other hand, we can see one nice qualitative thing. And that is that the elbow, the point at which the EEG transitions from a decrease to a saturation, seems to be around where the subjects transition from saying mostly short to mostly long. That is the bisection point. Now, this qualitative correlation encouraged us to think whether we can come up with a model that will give us a quantitative relation between the EEG on the one hand and the behavior on the other hand using a single cognitive process. And the model we came up with is as follows, and it's based on a model that was developed by Fuad Balchi, who was here last month, and Patrick Simon, who was here not long ago either. And the basis of this model is a DDM, a drift diffusion model, and a threshold. So the model works as follows. At the beginning of the stimulus, we have an accumulation process that starts. Model does a drift diffusion process. It accumulates noisily some evidence, which we can take as an internal representation of elapsed duration. Now, if the process reaches the threshold during the stimulus duration, this stimulus is categorized as long. However, if the stimulus ends before the process reached the threshold, then this stimulus is categorized as short. Now, using this simple model, we can predict both behavior and EEG. And we'll start with behavior. So let's take a look at the responses of the model across many trials. In blue, we have the mean and dispersion of the accumulated evidence across time. And in red, we have the probability of the process reaching the threshold after t seconds. Now, to learn what is the probability that a certain stimulus duration will be categorized as long, we simply need to take a look at the cumulative probability of the model reaching the threshold after so many seconds. This is equal to taking the cumulative distribution function for this probability density function, which gives us the psychrometric curve on the right here. So we have a transition from always saying short to always saying long. This fits qualitatively the behavioral responses in this task. Now, to look at the EEG, we wanted to take a more quantitative measure from the model because this one is just binary, short or long. So let's say that the stimulus was over, let's say after half a second, and the process did not reach the threshold. So there is some distance from the threshold. Now, on the other hand, if the process did reach the threshold during the stimulus, there is no distance from the threshold. So what we thought is that at the offset of the stimulus, the model basically emits a response that is equal to the distance from the threshold. Now, if we take a look across many trials, we have those printed on the bottom here. In blue, we have the distribution of positions of this process at the end of a stimulus, let's say half a second long or about 1 second long or so on. And in red, we have the mean distance from the threshold. So as the stimulus becomes longer, the distance decreases naturally. Now, we can do that analysis for all intervals in our block and anything in between. And we get the following function, which resembles the ERP quite a lot. So we start with a maximal response for the shortest duration, decreasing linearly, reaching an elbow, and a saturation for the longer stimuli in the task. Now, qualitatively, this matches what we saw, but now, of course, we wanted to get a quantitative fit to the behavior and the EEG. So let's pull up the results from the experiment again. So I remind you, on the left, we have the ERP traces. Then in the center, we have the summarized ERP responses. And on the right, we have the behavioral responses. And what we did is we fit first the model to the behavioral responses. Let's see what we got. This is presented in the solid curve here. And we got an almost perfect fit of the group results using the model. Now we took this fitted model and we used it to predict the ERP responses. And let's plot them as well. And we got, again, an almost perfect fit of the ERP responses based on the behavior of the subjects. And we got an R-squared of 0.98 for this group of subjects. So we have a model that can that is based on a familiar process for timing that can predict both the behavior of the subjects as well as their ERP. That was great, and it brought two questions to mind for us. First, we wondered whether this signature generalizes across sensory modalities. And the reason is because if you think of the model, there seems to be nothing specifically sensory about it. It should be like general to all temporal decisions. So we wanted to test that. The second question that came to mind is that if you think about this process as inherently involving a decision, it seems like it should track decisions in different temporal ranges. Or in other words, it should be sensitive to the relative rather than absolute duration that we presented subjects with. So to test the first question, we ran a second experiment in which we used the same intervals and same design as the experiment I just showed you. But we used, instead of visual gratings as stimuli, we used vibro-tactile stimuli to the fingers of subjects. That is, we switched the modality. So let's see the results we got. These are the EEG traces to the offset of the stimulus. Again, in x-axis, we have this time from stimulus offset. In the y-axis, we have the potential across the same channels that we saw before. And the color coding is as we saw before. So we have in the lighter pink, the shortest duration, half a second long, and in purple, the longest duration, 2.1 seconds long. And again, we see a decrease in the positive potential that seems to saturate for longer durations. Now, we can, of course, summarize these responses just as we did before. So we took the average potential across the same time window, 300 to 500 milliseconds post-offset. And I'll plot them now in the center here. So in the x-axis, we have the stimulus duration. In the y, we have the summarized ERP response. And we see a linear decrease and an elbow and a saturation. So the ERP seems to match what we saw before. And of course, the behavior of the subject also matches what we saw before. So subjects can bisect temporally tactile as well as they do visual. And we got this psychrometric curve. Now what we wanted to do is test our model, whether it can fit these results as well. So we fit the model first to the behavioral bisection curve, and we got a perfect fit. Then we took a look at what the predictions of the model were for the ERPs. And again, we got a very nice fit to the results of the ERPs. with an R squared of 0.93. Okay, so to summarize what we saw so far, we have an answer to the first question. This signature, this neural signature, seems to generalize across sensory modalities, both for tactile and visual. This brings us to the second question, whether this neural signature will track relative rather than absolute duration. So we designed and ran another experiment. This time we returned to the visual modality using the same ratings, but now we divided the range that we used into two interval ranges, divided into two blocks, such that each individual in the task ran the bisection task twice. Once with a short intervals block between 0.2 seconds to 0.8, and once in a long intervals block between 1 second and 2 seconds. Let's take a look at the results of this experiment side by side for the two blocks. So, on the left, at the top, we have the EEG traces to the offset of the stimulus for the short block, that is 0.2 to 0.8 seconds. And at the bottom, we have the same EEG traces to the offset of the stimulus, but this time for the longer durations block. And what we see here is again the same type of responses that we saw before. A strong positive potential after the offset of the stimulus that decreases with increasing duration and seems to reach the threshold for the short block and for the long block. So we summarize these responses again by taking the average for this window, and we plot them here. And we can see the linear decrease and the saturation for both blocks clearly. Now, of course, we can take a look at the behavior, and as expected, subjects can do the bisection in both blocks just as well. For the longer block and for the short block, we get nice bisection curves. And the last question to ask is whether the model can fit these results as well. So again, we fit the model to the behavioral results and use them to predict the ERP responses. And let's see what we got. We got, again, very good fits to the behavior and almost perfect fits to the EEG. So for the short block, we got an R squared of 0.98, and for the long block, we got 0.97. So the model can tracks basically decisions both in the short intervals block as well as the long interval block. And to summarize that, we can answer the second question positively. That this neural signature does track decisions. It does track relative rather than absolute intervals. So to summarize what I showed you, we found a systematic relation between offset responses and stimulus duration. These offset responses, first, are not modality specific. We found them both for the visual and tactile modalities. And they are sensitive to relative rather than absolute duration. We found them both for sub-second and super-second temporal ranges. Now, both the ERPs that we found and the subject performance in the task can be explained by a drift diffusion model. And this fit supports, first, dynamic perspective on temporal decisions, that is, perspectives on temporal decision-making, which can take place in the dynamic fashion. Rather than just at the offset of the stimulus, subject can make the responses during the stimulus, which seems critical. Second, it shows the usefulness of accumulation to threshold theoretical perspectives in analyzing the EEG in temporal decision tasks. And we hope that we can use this now to continue and test other questions about timing and time perception using this handle. So I want to thank you for your attention. And I would also like to thank my lab. First and foremost, Professor Ayelet for guiding and supervising this work. And then a group of talented colleagues and friends from the lab who helped designing and running the experiments I just showed you. And of course, the funding agency for their help in making this project possible in the first place. So I thank you again for your attention, and I welcome any feedbacks, comments, or questions.
Thank you, Nir, for your interesting talk, very interesting experiments. We will have 10 minutes for questions. So you can, if you have any questions, you can just raise your virtual hand on Zoom, or you can put them in the chat button so I can read them for you and Nir can reply. Okay, I'm just quickly checking the virtual hands now, and I can see Pat Seaman first. If you would like just to turn off your camera and microphone so we can be more, interactive, feel free to do so. If not, well, it's up to you.
Should I ask my question?
Yes, of course. Go
Okay.
on.
Well, thank you, Nir. That was beautiful.
Thanks.
I had one quick question and then one, well, they're both questions. One is just technical. Slide 48 on your upper left corner, I think maybe the legend is wrong. I just want to make sure. Are those?
The legend. Oh, you're right.
Okay.
Okay. Thanks for noticing that.
So those are shorter time intervals.
Yes, it's between 0.2 to 0.8 seconds.
Okay, cool. And those are huge effects. That's really impressive. I wonder if one way to put it is that what you are seeing is a modulation of the P300 or P3 effect, just sort of reduced in amplitude when you've kind of already decided. Is that...
Yeah, thanks for this comment. This is something we are thinking about too as well, of course. So it looks like the modern perspectives on P3 or the CPP, which involve decision making. So yeah, we definitely think it's related. Thanks.
Okay. The next hand that I can see is Asaf Breska, if I'm correct. So you can take over and ask your question.
Thank you, thank you, Nir. Really very interesting. I like the approach a lot. And my question is, did you look at what the EEG looks like during the presentation of your bisection stimulus? And the reason I'd ask is that I'd expect to see buildup of the CNV potential, which has a very overlapping topography to the topography that you showed here. So I wonder how much of this might be due to resolution of the CNV after the midpoint has elapsed, right? If we assume that what people are doing is they're building some kind of reference of what's the bisection point, and then waiting to see whether the stimulus is going to be before that. And once that time elapses, they kind of know their decision, and then we'd expect the CNV to kind of return to baseline. So is it possible that some of these differences are actually these CNV differences, but when you do baseline correction, you see them as a positive modulation?
So yeah, thanks for the question. We definitely do see the CNVs during the stimuli presentation as well. So as to what you said, I think that if it was just a resolution, so I'd expect actually similar potentials to shorter as well as the longest durations in the block, because for both we have like the CNV at the upper ranges. Is that correct?
Yeah, right. This is why I'm curious on how you think these processes interact, because it could compress some of your later intervals.
Yeah, that's an interesting question. We'll have to take a closer look at it. It did bring a question to mind that I wonder if you have an idea about. So, like, if we take the the DDM approach to this like offset potential, so of course other people talked about the CPP, which is actually locked to the onset of other decision-making intervals. And if it was just this, so I would have expected to see like a climbing activity at the beginning, which is positive, which seems to be like completely the opposite of what we get in time decision. So yeah, I think that there is like some interplay between these two.
But you definitely see in your 0.2 to 0.8, you can see in the baseline that some of them kind of show this buildup, the end of the buildup of negative potential. So this is why I was curious.
Yeah, thanks. So I think that for the shortest duration in the short intervals block, we're actually getting some leakage from the onset responses to the, because it's like 200 milliseconds or 300 milliseconds. It's before the onset ERP is over. Yeah, thanks. Good point.
Thank you.
Thanks.
Okay, I will move to the chat right now. We have a couple of questions there. So Nir, the question is, what do the individual results look like? Did you double the range of the shorter intervals because you had to in order to get the full function? If so, what does that imply about timing in the two ranges?
So the second question is whether we tested 0.2 to 0.4, correct? That's what I understood from the question. We didn't test that. It's a good question. I mean, like this, classic Weber law will tell you that we should expect the same results in the 0.2 to 0.4 and the 1 to 2 seconds. I haven't checked that, so I don't know how to respond to this question. As for single subject results, this is something we are playing with. We haven't done a full quantitative analysis yet, but qualitatively it does seem like, let's say, by eyeballing, that at least half of the subjects have something that looks like this.
Okay, that question was from Charles Randy, and then I moved to the question from Jonathan Cannon. Do the distribution of ERP amplitudes over trials look unimodal or bimodal, up, down? Could you get a similar range of ERP amplitudes if the amplitude was higher for one decision and low for the other?
Okay, yeah, that's a good question. One answer to this question is that, of course, in addition to the analysis we show here, we also ran a mass univariate GLM analysis on the results, in which we included as predictors both the duration of the stimulus, the decision of the subject, and an interaction term. And all three predictors came out as significant. So it's not just a question of the duration of the stimulus or the decision that the subject made. It is actually a combination of both, which is captured in the model. And maybe another answer is that if you compare, maybe let's take a look at the first one, the left part of the range, in which we have like a very small difference in the percent of long responses, but we get a much larger change in the amplitudes. So I think that just trying to account for it by differences in the number of trials in which we responded short is not enough. But yeah, that's a good question. Definitely something that came up.
Thank you, Nir. We will ask one more question coming from the raising hands, and then we can move on to the next talk. If you still have questions for Nir, if you can put them in the chat or contact us after the talk, and we can get in touch with Nir. Yeah. So the question comes from Pierre Divanai, if I pronounce that correctly. Pierre, would you like to take over and ask your question?
Yes, please. Divani, by the way.
Thank you.
Sorry if you see me, I look disheveled because it's the West Coast and very, very early in the morning. So thank you. It was a very interesting talk. My question is very simple. Have you also explored the auditory dimension, auditory modality, which would be very important for instance, musical performances?
Mm-hmm. Yeah, thanks for the question. We started looking at the auditory modality like in behavior, but we don't have yet an EEG experiment in the auditory modality. That would definitely be interesting to take a look at. Thanks.
Thank you.
Okay. Thank you, Nir, again for your presentation. That was great, and a great discussion followed afterwards. Again, if you have more questions, you can put them in the chat, for example, and then we can pass them on to Nir if we have more time at the end. we can also discuss them. For now, we will have to move on to our next speaker, if that's okay with everyone. Okay, I will share my screen again. Okay, Nicolas shared his screen. That's fine. Okay, I had also prepared a small bio for Nir, but I was quite nervous for this talk, so I didn't manage to say a few words, but Nir presenting himself. So we have Nicolas Meirhaeghe. He's a postdoctoral fellow at the Institut de Neurosciences de la Timone in Marseille, in France. And he will give his talk now about the precise and adaptive neural mechanism for predictive temporal processing in the frontal cortex. Okay, I will stop sharing my screen so Nicolas can share his screen. And then another 10 minutes questions will follow at the end. Thank you.
Okay, can you all hear me and see the screen properly?
Yes, it looks fine.
Okay, great. So thank you very much for having me. So my name is Nicolas Meirhaeghe. I recently finished my PhD working with Meirhaeghe at MIT, and I'm now a postdoc in Marseille working on monkey physiology. And today I'm going to present some of the work that I did towards the end of my PhD related to predictive temporal processing. So to motivate sort of the main question that I will be discussing, I want to show you a video, and maybe some of you have already seen it. But what you will see is the soccer player, Cristiano Ronaldo, attempting to score a goal in complete darkness. So let me just run the video. So here's Ronaldo getting prepared to kick the ball from this corner. And at this point, they turn off all the lights. So the result is that the ball ends up in the cage. And so the interesting question is, what happened in darkness? So this is the part that we can't see. So obviously this is not a sort of a regular goal because he's using his shoulder, but something that is very, very interesting to observe is that even though Ronaldo has no visual sort of cues about the ball coming, you can see how he's making a step towards the back as if he knew that the ball was going to land there, and he starts cocking his shoulder and initiates the movement right at the time that the ball hits his shoulder. So I think what this video illustrates is the ability of humans and other animals to be able to predict stimuli based on our prior experiences. And one of the big questions that I tried to investigate during my PhD was how are specifically temporal expectations encoded in the brain. So just a few words about background. When we think about expectations, we can typically think of those as separated into three categories. You can expect the what, the where, and the when of a stimulus. So we already know quite a lot about the what and the where. The main empirical finding is that we have neurons in the brain that are tuned to specific features in the environment, let's say a particular location, and those neurons will start firing in anticipation of a stimulus if that stimulus is expected to appear, let's say, in that particular location. These sorts of observations have been well explained by the theory of predictive processing, which posits that the sensory inputs we receive are not processed in their raw form, but rather in terms of deviations from expectation. So those neural responses, which are sort of an anticipation of the presentation of a stimulus, are thought to be compared and subtracted to the actual neural response, which is elicited by when the stimulus actually comes on and is presented. The main challenge is that we know that neurons are typically tuned to spatial and categorical features. And in other words, the what and the where questions are well understood. However, it's much less clear how to extend those ideas to temporal processing, that is to say, the when. So to investigate this question, I used a task in monkeys called the ready, set, go task, and the task goes as follows. So we ask monkeys to fixate a central computer screen, and we will present two sequential flashes of light to them. The first flash is the ready flash, and after some time called the sample interval TS, comes the second flash, which is called the set flash. After the set, the animal has to wait some amount of time before initiating an eye movement towards the target, which can be located either left or right of the fixation point. Now, the goal of the monkey on every trial is to make sure that the time between the ready and the set, which we control experimentally, matches the time between the set and its own motor response. And to be more specific, the animal is rewarded maximally if the TP, which is the produced interval, matches the sample interval, and the reward linearly decreases with the relative error. Now, we use this task to manipulate temporal expectations by picking sample intervals which come from two different distributions. So there's the short distribution, here color-coded in red, and the long distribution color-coded in blue. Each distribution has 5 equidistant intervals which will be uniformly sampled across trials. And the idea is that we're going to rapidly switch between distributions in short blocks of trials. We indicate to the animal which distribution is used on a given trial by changing the color of the fixation point. If the fixation is red, it means we're using the short distribution. If the fixation is blue, we're using the long distribution. So in the context of this task, the question I wanted to investigate was how temporal expectations about the timing of the set in these two conditions are encoded in neural activity. And just as a note, we actually published 2 papers recently, and today I'm going to mostly focus on the results that were presented in the latest paper in 2021. But if you're interested, definitely you can dig into both papers. So before going into the neural data, let me show you the behavior of monkeys in this task. So the behavior to show you the behavior, I will plot on the x-axis the interval that we present to the animal, so the sample interval, and on the y-axis, the produced interval. Obviously, you should expect the data to roughly lie along the unity line, that is, if the monkey is doing the task. And that's basically what you see. However, you see some very interesting deviations within each condition away from unity line. And specifically, you see that at the edges of each distribution, the responses of the animal seem to be biased towards the mean of each distribution. And that is even more striking if you focus, for instance, on this single interval here at the overlap of the two distributions. You see that the animal is biased in opposite directions depending on its prior expectation. So these biases indicate to us that animals do rely on their temporal expectations to solve this task, which means then that we can go into the brain and try to find a neural correlate of those expectations. So where do we go in the brain to find this neural correlate? So we record from a region called the dorsomedial frontal cortex, which comprises the pre-SMA and SEF areas, which are known to be implicated in incorporating prior experiences into timing behavior. And for the electrophysiologist in the crowd, the setup we used was a set of 3 linear probes with 24 channels each, which we inserted acutely and allowed us to record, let's say, from 100 neurons on a good day. So now I'm going to show you neural activity during this task, specifically in the measurement epoch. So that is to say between the ready and the set. And this is really the period where the animal is supposed to be waiting for the set flash to come. Here are 6 example neurons On the x-axis is the time aligned to the ready flash. And on the y-axis is the firing rate of each of those neurons. And the color code is going to be always the same, red for the short condition and blue for the long condition. So as you can see, neurons do have very heterogeneous profiles during this epoch of the task. But what you can already observe is that the patterns of activity of these neurons seem to be related if you compare the short and the long condition. Specifically, it seems like the pattern of activity in the long condition is simply a rescaled version of the pattern of activity in the short condition. So to get a better sense of what happens at the population level, here I'm going to summarize the activity of all the neurons that we record from. And I'm going to summarize it in a high dimensional space where each dimension will represent the activity of a single neuron. So if I isolate a single time point here, and I look at the activity across all my neurons at this time, this gives me a single dot in this high dimensional space. If I move to a different time point, I get a different state. And now if I start spanning the entire measurement epoch, I recover what is called the neural trajectory. Here are the two empirical neural trajectories associated with the two different conditions. So red for short and blue for long. What you can already see from this visualization is that the trajectories are highly similar across the two conditions, but you can see that the speed at which the trajectory evolves is much faster in the short condition compared to the long condition. And here you can infer the speed visually by looking at the spacing between consecutive states, which are separated by 20 millisecond bins. So how could speed differences in neural dynamics be related to temporal expectations? Our main hypothesis was that the neural speed might in fact encode the mean interval within each distribution. And here is what we were thinking specifically. Imagine the activity of the population here is summarized by this black dot. And the activity starts from a given state at the time of ready and moves towards the target state at the time of set, represented by this dashed line. Now, if the speed here is calibrated such that you reach the dashed line at the mean interval for the short distribution, now you can use the same logic for the long condition by simply modulating the speed, which then allows you to end up exactly in the same state now at the mean of the long distribution. The reason why this hypothesis was appealing to us is that what it allows the system to do is to encode each individual interval within each distribution as a deviation from the mean. That is to say, we have an invariant representation of the interval, which is not an absolute measurement, but now a relative measurement relative to the mean, which is consistent with this idea of a predictive process. The other reason why this hypothesis is appealing is that it makes a specific prediction, which we can test in the data. This prediction is that the speed in the short condition should be proportional to the speed in the long condition. And the coefficient of proportionality here, which I'm going to call the scaling factor lambda, should be equal to the ratio of the two means. So now we can test this prediction, in the data directly. For that, we need to be able to measure speed differences in the neural data. To do that, I applied the following technique. I basically ask, what is the time necessary to travel a certain distance along each trajectory? And for each condition, I'm going to call this time t short for the short condition and t long for the long condition. And here I'm going to plot the t long versus t short. And so for a single distance, I get a point on this plot. If I take a longer distance, I get a new point here. And if I span the entire trajectory, what I obtain is a mapping, a temporal mapping between the time in the long condition versus the time in the short. And what is pretty neat about this mapping is that the slope of this line is exactly equal to the coefficient of proportionality between the short and the long conditions. just to make it clear, if we are in a situation where the long condition is going slower compared to the short condition, we expect this line to be above the unity line and the scaling factor to be greater than 1. If the speeds are equal, then the line should be exactly on the unity line. And if this short condition is slower, now this line should be below the unity line. In our case, we had a strong prediction, which was that not only should the mapping be above the unity line, but the slope should be quantitatively close to the ratio of the two means. So here is the empirical temporal mapping that I obtained from the data, plotting the time in the long versus the time in the short. As you can see here, the prediction is the red line, and the prediction is essentially just a line whose slope is constrained to be equal to the ratio of the two means. And this result was robust across the two animals that we tested. So we concluded that here, the speed of neural dynamics quantitatively reflects the average time interval that the animals expect in each condition. Just as a side note, we performed an important control to make sure that these speed differences reflected expectations and not simply model preparation. Indeed, if you think about the design of the task, in the original paradigm, there is one hypothesis, which is the one that I've been exploring, which is that the speed during the ready set epoch reflects the mean of the sample distribution that we use. However, we could also think of an alternative hypothesis, which is that the speed during the ready set, in fact, reflects the mean of the produced distribution. So in that case, the speed differences would be more related to model planning and not expectation. So we used a control experiment where we essentially matched the two sample distribution across the two conditions. But in one condition, we asked the animal to produce 1 time the interval that was measured. And in the second condition, we asked the monkey to multiply this measured interval by 1.5. And this allowed us to match the sample intervals, but still have different produced distributions. In this control experiment, we had the neural trajectories were also separated based on the condition. However, visually, we did not see any speed differences. And I did the same exact analysis of the temporal mapping that I explained before. And here you can see that the data lies along the unity line, which indicates that the speeds were equal across the two conditions. Okay, so moving on to the last part of my talk, now I want to ask the more difficult question of whether this finding generalizes to a non-stationary environment, which is when the animal has to update its expectations on the fly. So to test this, we did a third experiment where we first exposed the animal to a pre-distribution, here shown above, with 5 different intervals. And then at some point in the session, we didn't warn the animal, but we collapsed this distribution to a single interval, which was in this case equal to the longest interval. The reason why we went to a single interval at the end of the distribution was because we reasoned that before adaptation, the animal will be highly biased towards the mean of the pre-distribution, as I showed you before. However, at the end of adaptation, the animal should no longer be biased. And so, in other words, this design should allow us to have a behavioral marker of adaptation. So here is the behavior of one monkey which underwent this manipulation. So on the y-axis here is the interval that the animal produces as a function of trials on the x-axis. What you can see is that immediately after we transition, you see that the responses of the animal are highly biased towards the mean of the pre-distribution, which was expected. But after several hundreds of trials, you see that this bias gradually goes away, indicating that the animal is updating its expectations. Now we can look, because those changes in fact occur within a single recording session, we can compare the neural activity before and after we make the transition. So here are again 6 example neurons, where on the x-axis is again the time aligned to the ready flash, and on the y-axis is the firing rate. And now the color code is blue for pre-transition and orange for post-transition. And as you can see, comparing those two moments of the task, several neurons change their patterns of activity during the adaptation process. Now, of course, what we wanted to do was to quantify this change and to see whether it was quantitatively predictable from the changes in expectations that we expected. So I'm going to use the exact same temporal mapping method that I used before. In the first experiment, we had 2 different distributions, each with a different mean, and we expected the slope of this mapping to be equal to the ratio of the 2 means. So now in the adaptation, we again have a different mean for pre and post transition. So now the new prediction is that the slope should be equal to the ratio of those 2 values. Here is the empirical mapping for one monkey. And as you can see, the data is consistent with the prediction. And this was the case for both monkeys that we tested. So in other words, the neural changes that happen during this adaptation process, they quantitatively match the new expected interval. As a last note, you can also do this, you can sort of quantify the time course of neural adaptation by looking at how the slope of this mapping changes across trials. And this is what I'm showing here. On the y-axis will be the slope of this temporal mapping. So before the transition, you expect it to be close to 1 because nothing has changed yet. And after the transition, you should expect at some point the slope to reach its final value here. What we found was that very rapidly after the transition, the slope of the mapping was adjusted. And again, this was the case for both monkeys. And something that was very interesting and intriguing to us is that if you compare the behavior of the animal at very early on after the transition, when the neural activity has already adjusted, the behavior itself has not adapted yet, which indicates that the neural changes, which correspond to changes in expectation, precede the behavioral changes that are related to those changes. So as a summary, what I showed you today is that temporal expectations modulate the rate of change of neurons in the frontal cortex. The speed of neural dynamics quantitatively reflects the mean expected time interval. The neural speed reflects temporal expectations and not movement preparation, which is the control experiment, which I did have time to show. And finally, when we performed an adaptation experiment, we found that the neural changes corresponding to changes in expectations preceded the behavioral adjustments. So finally, I want to thank my supervisor, Meirhaeghe, and also Hansam Zan, who's a postdoc in the lab who helped me for many aspects of my PhD, in fact. So thank you very much for your attention, and I'm ready to answer any questions if there are any.
Thank you, Nicolas. That was a very interesting talk and great work of yours. I can see now that 3 participants raised their hands. Let me check their names. So I think Pierre raised his hand first. So Pierre, do you want to go first with your question?
I think you're muted if you're trying to speak.
Am I?
No, no, not you.
Okay. Yes, I think Pierre, you're muted, so we cannot hear you if you're asking your question. Yes.
Can you, okay, can you hear me? Okay, well, it's really very, very interesting. And incidentally, this is exactly how we speak and how we understand speech. We have a certain state that we, of the articulator that we understand, that we sense through our articulatory system. And if we sense a speed, if we can evaluate the speed of transition, then we can predict the endpoint, even if that endpoint is not reached, because in speech, the endpoint is very, very seldom reached. But still, we understand each other relatively well. This is interesting. I don't know whether you can see this, but there is a book that we published, Ronnie Kelly, Mohamed Mayati, and myself, in 2017. It was published by the Grouter, calling speech a dynamic process. And I recommend you take a look at it online, because it explains exactly that what is important is assessing a state first and assessing the speed of the transition. And that's how we produce speech, and that's how we understand speech as well.
Okay, thank you.
Thank you, Pierre, so much. Let's move on to the next question. It comes from Pat Simon.
Hi, thank you, Nicola. It was a great talk. Great Ronaldo video. It's a really good way to start. I had one quick question about the task, though. I wanted to make sure I understood before I forget. So it's ready, set, go. You tell the monkey a trial is beginning. That's ready. The time from ready to set is defined by some flash. And then you know that go ought to repeat that duration, except when you manipulated that. And then there's a second cue for go, right?
So there's no cue for go. The animal needs to decide when to initiate the movement so that the time between the set and the movement response matches the interval we present.
And the reward triangle gives them the feedback they need for that.
Exactly, yeah. So in other words, this is really a timing reproduction task where we give an interval, the animal has to reproduce it immediately after.
Yes. Okay, good, thank you.
Sorry if it wasn't.
Yeah, I just wanted to make sure. So my other question is basically about the PCA. So there's nothing special. Well, maybe there is something special, but 3 dimensions is just sort of visually interesting.
Right.
But I wonder if you collapse down to just PC1, is what you see essentially a drift diffusion process? Is it just a ramp?
Yeah, I mean, I think you could see it that way. So first of all, I just want to make it clear for everyone that the PC plot that I showed is only for visualization. All the analyses were done in the full state space. So there's no trick there. But your question is actually in the other direction that I was expecting. You're saying, why do I even need 3 dimensions? I only need 1.
Well, I'm just curious about the 1.
Yeah,
I mean,
yeah.
I don't know which ones are functionally, causally relevant, but
I mean, I think in a way you're correct that this is fundamentally a one-dimensional system because the latent variable here is time. So the time can be encoded along a single dimension. The tricky part, which I didn't have time to go into, and we describe in the first paper that we published, is that the trajectory is actually not really following just a single line. It actually is curving at some point. And the fact that it uses not just one dimension, but multiple dimensions is to encode the distribution itself with the variance. Everything I said about the speed here encodes the mean. But in fact, we have other interesting results with this task where we find that the trajectory encodes not just the mean, but the full distribution. If that kind of answers your question.
I'd want to talk more about that. That's interesting. I'm not sure I totally get it, but yeah. Okay, that's interesting. Thank you. Yeah.
Thank you, Pat, for your question. We have a couple more. The next one comes from Sonia Gruen, and then Asaf Breska.
Hello, my name is Gruen. I was working with your colleagues together, Nicola, with Björk Kilavik and Alexa Riele. And we had a task where a monkey has to make a movement. This was motor cortex, either after 600 or after 1200 milliseconds. And after some time, we changed the request that he had to make a movement after 900 or after 1200 milliseconds. And we looked at both cases for exact synchrony. We used the unitary event analysis, and we found that when the monkey was expecting at 600 milliseconds, the signal, we found synchronous events at that time. And when he relearned the new task for 900 milliseconds, he first showed also the synchronous events at 600 milliseconds, but later on with practice, it moved to 900 milliseconds. My question is, you are looking at firing rates and co-firing rates. Do you intend also to look at the final time scale?
What do you mean by final time scale?
Well, fine in terms of synchronous spiking or correlated spiking. We looked at 5 millisecond scale, and there was a clear effect.
Mm-hmm. Yeah, I have to admit, I didn't look into this at this scale. So I never really looked at, for instance, spiking correlations. That's an interesting point. Yeah, I wouldn't, yeah, I think there's plenty of room to explore. I really don't know what to expect there. Yeah.
Thank you.
Thank you, Sonia. And then we have another question from Asaf.
Thank you, Effie. Thanks, Nicolas. Really super interesting stuff. I have a question and a half, if you may. If we have time. So the first question, I think, is kind of like what Pat was asking before, but from a different perspective. So this kind of morphing that, if I understand, you find, like what you say, the slower dynamics in the longer distribution are kind of exactly what you'd expect if you think about pacemaker accumulator kind of conceptualization about an integrator that's kind of building up at the pace of how many pulses you remember should be the interval, right? So that kind of adjustment of ramping activity, because it's integrating across a pacemaker according to how long you think the interval, how many pulses is the interval going to be. So did you look at the entire, when you look at the entire distribution of neurons, not in the state space, the majority of them show just a difference in slope between the two conditions? So you showed in the beginning, but is it true that the majority of them is like what's contributing most to the PCA are neurons like the top, I don't know, 2?
Right. So thanks for asking this question, because I wish I had time to show this, and now I can because of your question. So I actually looked at the level of individual neurons, and I asked this question of, if there is a scaling between the two conditions, I should just be able to take the pattern of activity of let's say this neuron in red, and I should stretch it by some amount and try to fit the blue pattern, right? That's like the definition of scaling. And so what I did that across all the neurons, and what I find is that actually the distribution of scaling factors is bimodal. There's one peak at 1, which indicates that actually most neurons or some neurons are not scaling between the two conditions. And then you get this other peak, which is centered at the value, which is the ratio of the two means. And in fact, here are the two monkeys separately. You can see this bimodality. So to answer your question, it seems that it's really a subpopulation of neurons. Not all of them are doing it, but
is this sorry, go ahead.
And it's not a continuum, in the sense that you could imagine another model where each neuron is scaling to some extent, And on average, at the population level, it scales by the mean.
Yeah.
It doesn't seem to be the case. It seems like all the neurons are scaling according to the mean.
Yeah. I guess what I was curious about is whether the neurons that do scale are the neurons that show ramping activity, like only the neurons like the two, like you don't have a neuron that does like an oscillation and that oscillation is suddenly scaled.
Oh, you do. Yeah.
You do,
I
okay.
think you definitely, I mean, look at those neurons, for instance, right? Like they don't just ramp up. They have very, very heterogeneous profiles, and it might be very dependent on which area you're recording from. Some people see, ramping might be specific to some areas, and DMFC is highly heterogeneous. So yeah, it's not. I guess that's why the model, the drift diffusion model, is correct. But it's correct at the population level, not at the level of a single neuron.
Yeah, so this is actually my question. Are most of the single neurons showing these dynamics? All right, cool. This was too long, so I wouldn't ask my half question. I'll leave it for email. Thank you.
Yeah, thank you.
Thank you, Asaf. I will go for the last question, and then unfortunately we have to wrap up because we're running out of time. Really great discussion. So the last question comes from Lando Lab. Nicolas, they say hi. When you said that the second interval in the ready, set, go is only related to temporal expectation and not motor preparation, you are excluding some cognitive processes like motor imaginary as well.
So I think you're referring to the control experiment, which is here. I'm not sure I fully understand the question, but let me try to reread it.
Mm-hmm.
So I think the underlying assumption here, or question is that maybe the process which happens between the set and the go, which is a movement preparation, is the same as the ready set, because you can sort of hijack the motor system to make time measurements. I think this is the flavor of the question. And so the fact that we find in the adaptation experiment, we find that the neural activity adjusts more rapidly than the movement itself, seems to really indicate that there is some dissociation between the two processes, between the motor planning and the temporal expectations. Now, I wouldn't necessarily know where to put motor imagery in that category, but what I can tell you is that whatever happens between the ready set, even if it's motor imagery, it seems like it's not being updated at the same time as what the motor system is doing during the set go. And that's to me, that's a very interesting result, which we haven't fully understood. But there are some theories out there which posits that there's no reason to expect that the system which make predictions about the world and the system which acts upon the world would adjust at the same rate. And there's a theory by Jordan and Wimmelhart about inverse models and forward models, which actually predicts these types of dissociations. So I hope I was kind of answering the question. Sorry if I misinterpreted it.
Okay, thank you so much. That was very interesting discussion. Nir and Nicolas, thank you for presenting your work today to us. That's really great research. If you have more questions about the speakers, feel free to put them in the chat, and then we can share them with the speakers. Before we go, I would like to give you briefly some info about our next journal club. So it's scheduled for 26th of January in 2022 with the new year. So we're hosting Dr. Andre Cravo. He is an associate professor in the Center of Mathematics, Computing and Cognition at Federal University of ABC. He's at the Timing and Cognition Lab, and we will share more information shortly. And as soon as we know more, you will receive an email with all the information. So this is the upcoming event. Thank you all for joining us today. And again, Nir and Nicolas, thank you so much. That was very informative. So I'm wishing you all Happy New Year, and we will see each other soon in our next virtual club. Thank you so much.